Chapter 2 Cell, the Building Block of Life. Notes and Summary from Grade 9 Subject Science Book Exploration. Introduction. All living organisms are made up of cells. A cell is the basic structural and functional unit of life. Some organisms like bacteria and yeast have only one cell and are called unicellular, while plants, animals and humans have many cells and are called multicellular. In multicellular organisms, similar cells form tissues, tissues form organs, and organs work together in organ systems. Yet, the cell remains the basic unit of structure and function in every living organism. The chapter also connects the idea of cells to the origin of life. It explains that early protective membranes may have helped in the formation of the first cells. This shows why the cell boundary is so important in living systems. 2.1 How to study cells. Cells are usually too small to be seen with the naked eye because the human eye has a limited resolution. The chapter states that 2 points separated by about 0.1 mm can be seen as distinct by the unaided eye. Since most cells are smaller than this, microscopes are needed to study them. Important points. A microscope magnifies tiny objects so that they appear larger. Friends, this is the structure of a light microscope. Robert Hooke was the first person to observe cells in 1665. He looked at a thin slice of cork through a self-designed microscope and named the box-like compartments cells. In school laboratories, light microscopes are used. Scientists also use electron microscopes, which show much finer details of cells at the nanometer scale. So friends, this is the figure of electron micrograph of lower surface of a colocasia leaf showing stomata. Activity-based understanding. The chapter explains how the size of a cell can be estimated using the field of view of a microscope. For example, if the visible field is 5000 micrometer and 25 cells fit across it, then the size of one cell is about 200 micrometer. This teaches students that microscopes are not only for seeing cells but also for estimating their size. Key ideas. Magnification makes the object appear bigger. Resolution improves clarity. Contrast helps distinguish parts of the object. So friends, this is a figure showing size of the objects and its visibility through unaided to aided eye. 2.2 Structure of a cell. A cell must interact with its surroundings and with other cells. These interactions take place through the cell boundary. 2.2.1 Cell membrane, the universal feature of a cell. The cell membrane or plasma membrane is a thin boundary that surrounds the cell and protects its contents. It gives the cell its individuality. Features of the cell membrane. It is selectively permeable. It allows some substances to pass through while blocking others. It controls exchange of materials between the cell and its environment. Osmosis. The potato experiment in the chapter shows how water moves through the cell membrane. In plain water, the potato swells. In concentrated salt or sugar solution, the potato shrinks. This happens due to osmosis, which is the movement of water through a selectively permeable membrane from a region of higher water concentration to lower water concentration. Diffusion and osmosis. Diffusion, movement of particles from higher concentration to lower concentration. Osmosis, diffusion of water across a selectively permeable membrane. Solutions around a cell. Isotonic solution, concentration outside is equals to concentration inside, like this. Hypotonic solution, outside is more dilute. Hypertonic solution, outside is more concentrated. So friends, this is isotonic solution, this is hypotonic, and this is hypertonic. Structure of the membrane. The chapter explains the fluid mosaic model. The membrane is made of a lipid bilayer. So friends, this is the lipid bilayer. Proteins are embedded in it. These are the proteins, various types of proteins. Lipids and proteins can move sideways. Proteins act like gatekeepers. 2.2.2 Cell wall, the outer covering of cells. Plant cells, fungal cells and bacterial cells have an additional outer layer called the cell wall. Functions of the cell wall: gives shape and rigidity, protects the cell, helps the plant remain upright, allows water and minerals to pass through because it is permeable. When plant cells are placed in concentrated sugar solution, the inner cell content shrinks, but the outer boundary remains unchanged because the rigid cell wall maintains the shape. In animal cells which do not have a cell wall, the cells shrink more easily. Composition: the plant cell wall is mainly made of cellulose, a carbohydrate. The chapter also mentions that cellulose in our diet acts as roughage. Plant cell versus animal cell. Plant cells fixed shape, rigid, have cell wall. Animal cells flexible, no cell wall, can change shape easily. So friends, this is a diagram showing a typical bacterial cell, A, this one, B, a typical plant cell, and C, a typical animal cell. 2.3 The cell interior, a coordinated working system. Most cells have three main parts: cell membrane, cytoplasm, nucleus. The cytoplasm is a jelly-like semi-fluid substance in which organelles are present. Prokaryotic and eukaryotic cells. The chapter compares bacterial, plant and animal cells. Prokaryotic cells. Pro means primitive and karyon means nucleus. No well-defined nucleus. No membrane-bound organelles. Genetic material lies in a region called nucleoid, usually smaller and unicellular. Eukaryotic cells, Eu means true and karyon means nucleus, have a well-defined nucleus, have membrane-bound organelles, larger and more complex, can be unicellular or multicellular. Comparison: prokaryotic cell size about 1 to 10 micrometer, eukaryotic cell size about 10 to 100 micrometer. The chapter also mentions cytoskeleton and cell inclusions in eukaryotic cells. So friends, this is the cell membrane, this is the nucleus, this one, this is endoplasmic reticulum, this is Golgi body, this is mitochondria, this is cytoplasm, this is ribosome, lysosome, vacuole, this is the vacuole in plant cell, this is chloroplast, this is cell wall in green, this is ribosome. Here this is ribosome, this is nucleoid genetic material, cytoplasm, and this is appendage for locomotion. 2.3.1 Why do eukaryotic cells need these organelles? Eukaryotic cells perform many life processes at the same time. To manage this, they have different organelles that work like different departments of a factory. Some organelles make proteins, some store materials, some package products, and some produce energy. Nucleus, house of coded instructions. So friends, this is the structure of a nucleus. This is nucleus in purple. This is nucleolus. This is nuclear pore. This is chromatin. And this is double layered nuclear membrane. The nucleus controls the activities of the cell. Structure: covered by a double layered nuclear membrane, has nuclear pores for transfer of materials, contains nucleolus, contains chromatin and chromosomes, functions: stores genetic information, controls cell activities, helps in inheritance of characters. DNA, genes, chromatin, chromosomes. DNA carries genetic information. Functional segments of DNA are called genes. In a non-dividing cell, DNA is present as chromatin. Before division, chromatin condenses into chromosomes. The chapter also notes that mature human red blood cells do not have a nucleus. which allows more space for hemoglobin. So friends, this is the diagram showing from cell to a DNA here. Ribosomes, the protein factories. So friends, this is the ribosome. This is rough endoplasmic reticulum and this is smooth endoplasmic reticulum. Ribosomes are tiny structures found freely in the cytoplasm or attached to the endoplasmic reticulum. Function, they are the sites of protein synthesis. Endoplasmic reticulum ER, manufacturing factory. So friends, this is the diagram, endoplasmic reticulum and Golgi apparatus, pathway for protein processing and secretion. This is the plasma membrane, this complete is Golgi apparatus, this is vesicles. The ER is a network-like organelle spread through the cytoplasm and connected to the outer nuclear membrane. Types of ER, rough endoplasmic reticulum RER, this one, has ribosomes attached, helps in protein synthesis and protein secretion. Smooth endoplasmic reticulum SER, this one, has no ribosomes, helps in synthesis and storage of fats and hormones. Golgi apparatus, the packaging and shipping centers. The Golgi apparatus is made of stacks of flattened sacs like this. Functions, modifies proteins and lipids, sorts them, packages them into vesicles, sends them for transport or secretion, helps in lysosome formation. It acts like the post office of the cell. So friends, these are the vesicles. Lysosomes, the cleanup system. Lysosomes are single membrane sacs filled with digestive enzymes. Functions, break down unwanted proteins, fats and carbohydrates, digest damaged organelles, remove waste, keep the cell clean and healthy. The useful products released after digestion may be reused by the cell. Mitochondria, the powerhouse of the cell. So friends, this is the structure of mitochondrion. This is the DNA. This is intermembrane space. These are the ribosomes. This is cristae. This is inner membrane and this is outer membrane. Mitochondria produce energy for cellular activities. Structure, double membrane. Outer membrane is smooth. Inner membrane is folded into cristae like this. Function, site of cellular respiration, releases energy from food molecules, stores energy as ATP, adenosine triphosphate, the energy currency of the cell. Special feature, mitochondria have their own DNA and ribosomes, suggesting an evolutionary link with bacteria. Plastids, center for food synthesis in plant cells and beyond. Plastids are found mainly in plant cells. Chloroplast. So friends, this is the diagram, structure of a chloroplast. This is the inner membrane, this is outer membrane, this is DNA. These are the ribosomes and this is stroma. Green plastids contain chlorophyll, carry out photosynthesis, have double membrane, contain stroma, also have their own DNA and ribosomes. Chromoplast, these plastids contain pigments other than chlorophyll. Function, give flowers, fruits and some vegetables their yellow, orange or red colors. Help attract pollinators and seed dispersing animals. Leucoplast, colorless plastids, store food such as starch, oils or proteins found in storage organs like potato and taro. How do flowers, fruits and vegetables acquire varied colors? They get their bright colors mainly from chloroplast, which contain pigments other than chlorophyll. These colors are useful because they attract insects and animals for pollination and seed dispersal. Vacuoles, the organelles for storage and support. A vacuole is a membrane-bound sac used for storage. In plant cells, usually one large central vacuole filled with cell sap, stores water, minerals, sugars and waste, maintains internal pressure and keeps the cell firm. When plants lose water, the vacuole shrinks, cells become less firm and the plants wilts. In animal cells, vacuoles may be present, usually smaller, help in temporary storage. 2.4 How do normal cells grow and divide? Living organisms grow because their cells divide and form new cells. Cells do not grow endlessly in size. Instead, growth occurs mainly through cell division. This also helps in repair of damaged tissues, replacement of worn out cells, reproduction. The onion root tip activity shows different stages of cell division because cells there divide continuously. So friends, this is the diagram showing different stages of cell division on onion root tip cells. 2.4.1 Cell division. Cell division is the process by which new cells are formed from pre-existing cells. Importance: growth, repair, maintenance, reproduction. There are two major types of cell division. Mitosis. So friends, this is the diagram showing mitosis is the process that produces two genetically identical daughter cells. Mitosis is the most common type of cell division. Features: one parent cell divides to form two daughter cells. So friends, this is the one parent cell and these are the two daughter cells. Daughter cells are genetically identical. Same number of chromosomes as parent cell. Importance: body growth, tissue repair, maintenance, asexual reproduction. Next, meiosis. This is the diagram. Meiosis is a two-step process that produces four gametes. Meiosis is a special kind of cell division that occurs in reproductive cells. Features: one parent cell divides twice. So this is the parent cell. Here it divides first into two, then it divides twice here into four. Produces four daughter cells. These are the four daughter cells. Each daughter cell has half the number of chromosomes. Here there are two chromosomes. There is only one and there is only one. Produces gametes such as sperm and egg. Importance: sexual reproduction. Creates variation and diversity among organisms. Occurrence: in humans, testes and ovaries. In plants, anthers and ovaries. Errors in division. The chapter explains that improper mitosis may lead to tumors and abnormal cell growth, while errors in meiosis can lead to genetic disorders, developmental problems, pregnancy loss, or reduced fertility. 2.5 Cell theory, the unifying principle of biology. The cell theory was developed through the work of Matthias Schleiden. All plants are made of cells. Theodor Schwann. All animals are made of cells. Rudolf Virchow. All cells arise from pre-existing cells. Classical cell theory states: 1. All living organisms are made up of one or more cells. 2. The cell is the basic unit of structure and function in living beings. 3. All cells arise from pre-existing cells. This theory unifies biology because it applies to all living beings from bacteria to humans. 2.5.1 Do cells grow and reproduce forever? No, cells do not live forever. Normal cell behavior: grow in a controlled way, perform specific functions, die when no longer needed, are replaced by new cells. Contact inhibition: many animal cells stop dividing when they come in contact with neighboring cells. This is called contact inhibition. Cancer cells: cancer cells lose this control and continue dividing uncontrollably, forming tumors. Some tumors may spread to other body parts. The chapter also mentions programmed cell death, PCD, an important process that helps maintain balance in the body and is essential in development, such as forming fingers in an embryo. Summary of the chapter: the chapter explains that the cell is the building block of life and the basic structural and functional unit of all living organisms. Cell may be unicellular or multicellular, but in every case they perform the essential functions of life. Because cells are too small to be seen with the naked eye, microscopes are used to study them. Robert Hooke first observed cells, and later improvements in light and electron microscopes helped scientists understand cell structure better. Every cell is surrounded by a cell membrane which is selectively permeable and controls the movement of substances. Plant, fungal and bacterial cells also have a cell wall which gives them shape, support and protection. Inside the cell, the cytoplasm contains many organelles that work together like a coordinated system. The nucleus controls cell activities and contains DNA, genes, chromatin and chromosomes. Ribosomes make proteins. ER helps in manufacturing substances. Golgi apparatus packages and transports materials. Lysosomes digest waste. Mitochondria release energy. Plastids help in food synthesis and storage. And vacuoles store materials and maintain firmness in plant cells. The chapter also explains the difference between prokaryotic and eukaryotic cells. Prokaryotic cells are simpler and lack a true nucleus and membrane-bound organelles, while eukaryotic cells are more complex and highly organized. Cell division is another major idea in the chapter. Mitosis produces two identical daughter cells for growth and repair, while meiosis produces four gametes with half the chromosome number for sexual reproduction and variation. Finally, the chapter presents the cell theory, which states that all living organisms are made of cells. The cell is the basic unit of life, and all cells arise from pre-existing cells. So friends, we have completed notes and summary of chapter 2, Cell, the building block of life. We will do important and difficult keywords with definition of this chapter and MCQs and extra question answers also in a separate video. If you like the video, kindly like, hype the video and share it with your friends. Also subscribe to my channel and press the bell icon to receive new video notifications. We will meet in the next video. Thank you for watching.